welcome to our Frontiers lecture. So I'm excited to announce that today's speaker is Dr. Roy Dar. He has a very impressive resume. Dr. Dar is an assistant professor of bioengineering here at the University of Illinois, as well as an affiliate faculty with the Department of Electrical and Computer Engineering, the Carl R. Woese Institute for Genomic Biology, the Cancer Center at Illinois, and the Center for Biophysics and Quantitative Biology. Dr. Dar received his bachelor's in physics and mathematics from the Hebrew University of Jerusalem, Israel, his PhD in physics from the University of Tennessee in Knoxville, and was a postdoctoral fellow at the Gladstone Institute at the University of California, San Francisco. His research investigates the use of gene expression fluctuations or biological noise to understand the control biological systems. So without further ado, I'll let him talk about the rest. Dr. Roy Dar.
Thank you. Thank you. Great. Well, thank you for the introduction and to the organizers and for the invitation to be here and present research from my lab. I'm very excited to share some of the results that we have and actually try and make a connection to brain somewhere in the talk, so it will connect to the seminar series. I also have my email address on the opening slide, so anybody that wants to follow up with some afterthoughts or discussions or questions is invited to do so. So my lab is a noise biology lab in that we aspire to understand and control fluctuations in gene expression to gain desired biological functions at the single cell level. So here you're looking at a movie of T cells that are adhered to a glass substrate, and they're being imaged over time, and they're expressing a fluorescent protein. And these are some of the types of technologies that we're using in order to measure single cell measurements. We also use computational and systems biology to understand, other than looking at simple gene circuits in single cells, we're also interested at organizational principles and genome-wide phenomena in order to reverse engineer complex natural systems. So we're kind of taking multi-pronged approaches to engineering the cell and biasing trait decisions at the single cell level. So starting at a 30,000-foot level, we can ask the question that if our engineering forefathers designed in a predictable framework, will the same be said about us? So so the silicon wafers that integrated circuits are integrated upon, and components are placed upon at different locations on this silicon wafer, you have a known input and output of that component. And that's because the material substrate and environment are homogeneous, static, and controlled. We know, though, that on the other hand, that the DNA and a virus that integrates at different locations in the DNA will have different outputs, even though the genetic code of that virus is the same. And that's because the biological substrate and its environment are heterogeneous and dynamic. So these are the sort of questions that we're thinking about, and specifically we're thinking about this in terms of noise. What is biological noise? Biological noise is due to the molecular crowding that causes random timing in biochemical processes. So even though we think of biochemical processes perhaps occurring in an aquarium or in void, it doesn't do so. It actually occurs in a very crowded environment. David Goodsell depicts here illustrates the E. coli cross-section. And if you have an enzyme somewhere on the outskirts here that's trying to get to its substrate, it takes a very convoluted path to get there. And every time it gets to that substrate, it'll create a protein product, or a product. Now, every protein that's being expressed in the cell has fluctuations that are due to this crowded environment, and that's what we're calling noise. So fluctuations in gene expression are caused by the random timing and discrete nature of molecular interactions that occur during transcription, translation, and degradation processes of both mRNA and protein. And there's also global resources that are fluctuating in the cell. So shared resources such as ATP, amino acids, ribosomes, RNA polymerase, they're all fluctuating as well. And they couple into the gene expression fluctuations of all the genes within all the gene expression that's occurring within our cell. So if we look at a specific gene circuit, in this case, I'm showing you the T cells again. In this, they have the HIV LTR promoter driving a green fluorescent protein, and it's a clonal population that's been grown over many days under microscope. Then you can see that two cells that are in the same vicinity of each other can have vastly different expression levels. So cell-to-cell variability in the protein levels is high. We can monitor noise in HIV by looking, for example, here I'm showing you a clonal cell population that has two HIV promoters. One is driving a green fluorescent protein, the other is driving a red fluorescent protein. So this is only HIV's promoter, not the full-length virus. Now, if these cells are adhered to a glass substrate with liquid media above them, and they have, they're imaged invertedly. And what you're looking at in the movie is a 600 by 600 micron imaging view, and the z-axis is time. It's about 20 hours. And what you can see is if both promoters are firing about equivalently, you would get yellow. So you have a lot of cells that are yellow, but more interestingly is you have some cells that are only red or only green. This is suggesting that the promoters are episodic in their transcription or in their expression levels. So it's highlighting that the viral promoter has bursts of expression, and that these bursts of expression have implications for the full-length HIV construct. The full-length HIV construct, which I'll get into later, is driven by this promoter, and the decision of whether it turns on or off is determined by these sorts of fluctuations that I'm showing you here. So back to this story of noise biology and understanding stochasticity, we can think about stochastic design of biasing the cell fate distribution of a population of cells as a Plinko board, where each ball on the Plinko board is a cell, and the population of cells at the bottom is the cell fate distribution, and the pegs of the board, their patterning, their composition, is defined by the cell environment, cell signaling, and gene regulation. And this would be the fundamentals of noise. What we're interested in are the applications of modulating the board, using synthetic and systems biology, pharmaceuticals, nanobiotechnology, perhaps biomaterials, in order to rearrange the board and redistribute the cell fate of the population between, for example, an active replicating virus and a virus that's latent or inactive in its fate. So a phenotypic switch in the population. So we work on HIV. We also work on stem cells. Those will be the two parts of the talk. HIV is a global epidemic. Currently in 2021, we're actually at over 38 million people worldwide that are HIV positive, a majority of which are in sub-Saharan Africa and Southeast Asia, regions of the world that don't have access to antiretroviral therapy. The promise of highly active antiretroviral therapy so antiretroviral therapy is a cocktail of drugs that inhibit the viral life cycle. So it inhibits the viral life cycle at various stages of the viral life cycle, such that if you have a patient or an HIV positive individual, that their viral RNA is at some level, and they go on HAART, then their RNA in their blood plummets below a limit of detection. And you would think that they're cured of the virus because you think that they don't have any viral RNA. But the minute they either stop taking the medication on purpose or not on purpose, the virus actually rebounds back to original levels of viremia within 2 to 4 weeks. So there's some ongoing active, spontaneous activation of virus that's occurring below the limit of detection, even though you're not detecting virus in the blood, RNA in the blood. And this occurs because HIV evades drug cocktail therapies through alternate fates of integration. Upon infection of CD4 positive T cells, HIV also infects other types of cells. I'll get to that later, other host cell types that are of interest. But in this case, CD4 positive T cells HIV primarily takes an actively replicating state where it hijacks the cell of its resources. It integrates its genome into the DNA of the host cell. It creates hundreds of viral progeny. It lyses the cell, and in a chain reaction, it infects all the other cells, all the other T cells. An alternate fate of integration is proviral latency. So the reservoir of cells that are inactive or dormant or quiescent, and they're non-expressive. And the problem with this latent reservoir is it can reactivate spontaneously at a later time point. So this arrow is the major barrier to a cure. It's the reason why that level of virus in the blood rebounds back the minute you take away HAART therapy. So there's two approaches to get rid of the latent reservoir. Either you shock and kill all these cells, you turn on all the cells out of latency into an actively replicating state with very potent drug cocktails. The other option is to actually stop this spontaneous reactivation from occurring and force these cells into a deep latent state. We call this block and lock strategy. So in this case, the cells would not turn on, and you could remove HAART. You could remove the highly active antiretroviral therapy, which has a lot of side effects. It's got cardiovascular diseases and neurological diseases that are associated with it, early onset of aging. Not everybody has access to HAART. So there's a lot of problems with antiretrovirals at the moment. So shock and kill, the first strategy to reactivate latent HIV, currently falls short. In patient experiments, only about 5% of the latent cell population can be purged. So here the yellow cell is the latently infected population. You would use very potent drug treatments, and you would purge the cell to be productively active, creating virus, and it would go through HIV-induced cell death. And in tandem, you would have HAART that would protect the bystander cells. Then at a later time point, you can remove HAART. So after all the latent cells have gone through cell death, you can remove HAART and the person would be HIV negative. That would be the hope. But it falls short. So this actually motivates us back to the stochastic design and noise modulation, and can we use theory from there in order to enhance the drug treatments that currently exist. So what we try to do is find noise modulating drugs and cocktails that enhance the ability to reactivate latent HIV, or to either to destabilize latency or to stabilize it more, so to control the stability of latency. So in order to do that, you first have to identify that there are two primary sources of noise that determine the stability of HIV latency. The first I alluded to with the movie earlier is that there's this bursty transcription. The HIV promoter can be looked at as an episodic transcription model of a two-state model where RNA polymerase is stalled right after the transcriptional start site, and it can go into an active state with with a train of RNA polymerases that rush through, create a burst of mRNAs, and then it can decay back into the off state. So you can use different drug treatments in order to modulate the pulse train or the promoter activity train of this of the transcription. Now these transcriptional bursts are amplified. These fluctuations at the transcriptional level are amplified by a master regulator of HIV. HIV is a 9-gene virus. It's only 9 genes. It's about 9 kb, and it's causing all of these problems. It's 9 genes that code for 15 proteins. The master regulator TAT, which is the transactivator of transcription, is a very potent positive feedback loop. It's a positive autoregulatory loop. So it amplifies fluctuations is basically what it does. But it's important from a noise aspect as well, and we'll see that later. So just to give you a little bit of perspective, what I'm trying to do with controlling bet-hedging circuitry through noise modulation is you can think about this conceptually as a chemical reaction efficiency where you can either use a catalyst to reduce, to deterministically lower the activation energy barrier on a potential energy landscape, or alternatively, you can introduce thermal fluctuations, for example, with a Bunsen burner, in order to get additional chemical reactions to occur. The analogous situation on the right is if you look at Waddington's epigenetic landscape, where a cell is represented by a ball or pebble at the top of the hill and can make various fate decisions or state transitions based on forks in the landscape. So small molecule treatments can cause enhancement of noise in gene expression and can further bias the cells in and out of certain states, state 1 or state 2, or in this case, latent HIV or active HIV. So can gene expression variability be used to control phenotypic switching of HIV? In this case, HIV is this untreated population, and it's at a lower expression state, and it has to pass some phenotypic threshold in order for that HAART master regulator to initiate positive feedback and to exit latency. So activators currently are only partially penetrating. They're only partially reactivating the virus. But what I'm suggesting, or what we're showing, is that if you introduce a variable drug that only changes the variance of the expression without changing the mean, and then when you combine it with the activator, you can get more threshold crossings and get more reactivation to occur. So you can see this here as well with stochastic simulations of a population of latent cells. So this is all with simulations over time. The gene product level here would be the master regulator, and they're all below the active replication threshold. And the untreated population, when an activator is added, then the promoter activity, there's an initiation of the promoter more often. You get partial penetrance. But with a noise enhancer, that the promoter activity would be less frequent but more intense bursts of mRNA. You can get the same mean expression level, but with a larger variance. And then in combination with the activator, the two together are predicted to give you more threshold crossings. So that's kind of the motivation behind the first noise drug screen that was ever performed, on HIV. And this was published, so it's a screen for noise of treatments that synergize reactivation of HIV latency. So this is a drug screen that modulates promoter expression variability and not mean expression level. We start with a T-cell population that has the HIV promoter, LTR, driving a destabilized green fluorescent protein, and it's plated in a 96-well plate format. Using automated liquid handling on an FDA small molecule library of 1,600 compounds were added to these 96-well plates for 24 hours of treatment. And then they were run on a flow cytometer with high throughput sampling system. So for those that are not familiar with flow cytometry, you use hydrodynamic focusing in order to focus cells into a single cell line, and they pass through a laser excitation source. There's an array of detectors that can collect the emission spectra and the intensity of the cells, and you can quantify the single cell fluorescence measurements that way. So here are some examples of noise enhancement. The purple lines are the noise modulators. The x-axis here is the mean green fluorescent protein for a normalized population of cells. The top two would be noise enhancers, and the bottom one would be an example of a noise suppressor. And this is a log axis, so these are real effects. So when we look at these 1,600 compounds, This is quantifying noise as the coefficient of variation squared. So it's the variance over the mean squared. The black squares are the untreated controls for each day of experimentation. The circles are each circle is a unique drug compound from the screen. And you can see that there are compounds that enhance the noise without changing the mean, or suppress the noise without changing the mean, and some that activate. So in total, there were 85 noise enhancers that were detected in the screen. And in addition to the noise enhancers that were detected in the screen, there were control compounds that were also tested. There were known chromatin modifiers. These are histone deacetylase inhibitors, methyltransferase inhibitors, and bromodomain protein inhibitors, that all these chromatin modifiers were shown to enhance noise of the promoter. And then there were known transcriptional activators of the LPR promoter through its NF-kappa B sites. These are PKC agonists such as TNF, prostratin, and PMA that decrease noise while increasing the mean. So they're activating the promoter. Now, the importance of this plot is that these are known compounds that are known to synergize reactivation of latency with each other. So these chromatin modifiers that are enhancing noise synergize reactivation of latency with these activators. They're already known to do that. So in a sense, this already corroborated the hypothesis. It already showed us that we're on the right track, because it's showing that noise enhancers and activators are synergizing. So we had the result based on the controls, but the next step was to ask how do the noise enhancers that we detected, these 85 noise enhancers, how do they perform on full-length latent HIV with an activator? Do they synergize reactivation? So can they be used in a cocktail drug treatment, a noise modulating cocktail treatment that further reactivates HIV? So that's what we did next. So instead of just looking at the promoter-only system, which I just showed you, now we're looking at full-length HIV virus. This has a deletion of the envelope, so it's replication incompetent, so we can work with it in the lab. It also has a green fluorescent protein in it, so we can detect it whether it's on or if it's off. And it's a clonal population, so it's a single integration site. On the y-axis, you're seeing the percent reactivation. On the x-axis, all 85 noise enhancers without activator, without tumor necrosis factor that activates the NF-kappa B sites in the LTR promoter. You have no reactivation of the virus or of the population of cells. And for all noise enhancers, you can see very few. There's just a couple of blips of purple that are showing that without activators, the noise enhancers by themselves do not reactivate latent HIV. But in combination with TNF, with the activator, which the activator alone gives about 10% on, in combination, about 75% of these drugs synergize reactivation of latency. So we saw an enrichment of synergizers, of reactivation of HIV latency among the noise enhancers. We went on to show that these drugs also work in a primary cell model of latency. What I showed you on the previous slide was in a T cell cell line. It was in a cancer cell line, a T lymphocyte. This is in CD4 positive T cells that are taken from an HIV negative donor's blood and then infected with HIV, full-length virus. And this is in collaboration with Bob Silicano's lab at Johns Hopkins Medical Institute. You can see that the percent reactivation of no treatment or the noise enhancers by themselves give very little reactivation. With activator, you get the blue bar, which is at about 65% on. And then in combination with the noise enhancers, you see that a majority of them are synergizing reactivation of latency. So
we were able to show that these noise enhancers also worked in primary cell models. We also did some optimization. We did dose responses of the noise enhancers to show that we get further reactivation. We did combinations of 2 activators with the noise enhancer or 2 noise enhancers with an activator, and we showed that all these different cocktails can further destabilize latency. But what about the opposite strategy? I mentioned earlier that there's, other than trying to shock latency and destabilize it, there's the opposite approach of forcing it into a deep latent state called block and lock. You can use noise suppression to actually pull down an endogenous reactivation event, signaling event that would destabilize latency. And you can see that here in the simulations, and it works out in the theory as well, that an activator with a noise suppressor would pull down reactivation events. And out of the 9 noise suppressors that we detected, one of them actually suppressed latent reactivation in both cell lines that I'm not showing you, but also in the primary cell model of latency. Here you're seeing prostratin, which reactivates to about 60%, is cut down by about half with S1, with the combination of S1, one of the noise suppressors. Okay, so this noise drug screen used flow cytometry, which is capturing a single time point after drug treatment. But we know that this noise is dynamic. We know that gene expression has fluctuations that are time dependent as well. And that's what I'm showing you here in these simulations. So we didn't capture the dynamic nature of the noise or the frequency components in the noise. In addition, I mentioned in the beginning of the talk that there were 2 noise sources of interest in HIV. There's the promoter and there's the feedback loop. And we only looked at the promoter in this drug screen. So this is where Yang Liu, a PhD student in my lab, took the lead and spearheaded a project that was published this year in PNAS. He did a noise drug screen for the dynamics of treatments that control HIV latency and its stability. So in his drug screen, what he did is, instead of looking at the promoter only, which you see on the left, now his construct that were in T cells was the HIV LTR with the destabilized green fluorescent protein driving an IRES, an internal ribosomal entry site, and TAT, the transactivator of transcription. So it's a mini feedback loop in a clonal active T cell population. So this is not a latent clone of HIV, it's a mini virus that we're trying to measure the noise of it. And the way that we're measuring the noise now is, instead of using flow cytometry, we use time-lapse fluorescence microscopy, automated fluorescence microscopy. So this has an environmental chamber and XY stage that it's fully motorized and autofocus and everything that's needed. In his case, in this drug screen, we screened over 1,800 compounds from a National Cancer Institute diversity library, and we're able to capture, in addition to the noise magnitude, we're also capturing dynamics, or the correlation time. So in addition to positive and negative controls on a 384-well plate, all of these cells are now imaged, adhered to a glass substrate with liquid media on top, and they're screened for 48 hours in 15-minute intervals. You can see here the constellation of T cells as the microscope, a 10x objective, is scanning and imaging every well that has a unique drug compound that's treating the cells. So the single cell signal extraction, we have to do segmentation and tracking of all the cells in all of the wells, but for each well and each treatment, we're measuring the mean fluorescence as a function of time for various single cells. So you can see here an example, just a sample of what we're doing. And then this would be this cloud of fluctuating signals is for one well on a 384 well plate, and each trajectory that's fluctuating is a single cell over time. So this is an 18-hour trajectory of its intensity. We then can analyze and map a noise coordinate for each compound. So we do signal processing, and we extract the stochastic component from the deterministic component. So we calculate the noise in gene expression, and then we calculate noise autocorrelation functions, which basically the autocorrelation function is a Fourier pair of the power spectral density. So it's giving you, it's probing the frequency domain of these signals. So we can calculate the population-based autocorrelation function and extract the half correlation time, which we call tau half. It's the time at which the signal has decayed, this autocorrelation function has decayed to half of its zero lag value, or half of its variance. So each coordinate is made up of the mean expression level, the noise magnitude, and the dynamics, or the half autocorrelation time. So this is what the noise map looks like now. So before we saw a noise map that was two-dimensional in the first noise drug screen. Now this noise drug screen is three-dimensional. It's made up of two-dimensional, two plots, noise magnitude versus mean expression level, and noise autocorrelation time versus mean expression level. The black squares are the untreated controls for each day of experiment. The controls are shown here. You have positive and negative controls that either up-regulate or down-regulate this miniature virus that we're screening. And then, more importantly, are the circles. The circles are the unique compounds, and if you take plus minus 2 sigma as our metric for selecting the hits, you can see in red are the hits that were noise modulating compounds for noise magnitude, or the red circles are the compounds that were noise modulating compounds for noise autocorrelation. So these are noise modulating compounds that are now changing the dynamics of the gene expression fluctuation, as well as the noise magnitude. So the next step, again, just like the first screen, was to test, is there an enrichment of either stabilizers or destabilizers of HIV, of full-length HIV latency with our noise modulators from this screen? And the answer is yes. What we did is we looked at the full-length virus again with flow cytometry. This is 2 rows where each row is a different clone of full-length HIV of latency. One is JLAT 9.2. The other one is JLAT 15.4. These are 2 unique integration sites. These are populations of latent HIV. The y-axis is the percent reactivation. The x-axis are the different noise modulating compounds. TNF is the red bars are 3 different days of experiment and their reactivation levels. It's the activator. So the activator alone are the dashed lines on each day of experiment, and you can see that the orange bars are all the synergizers that we got. So we got 34 out of 115 noise modulators that were assayed were latency reversal agent synergizers. And more importantly, I mentioned in the first drug screen that we only found 1 latency promoting agent, one of those stabilizers that suppress reactivation of latency. And in this screen, we found, according to these 3 blue arrows that I'm showing you here, 3 latency promoting agents where you can see that the bars are below the dashed lines. So they consistently, D35, D75, and D106 of these compounds consistently lowered the reactivation. And why is this an enrichment of either synergizers or stabilizers of reactivation of latency? Because when we take a random set of compounds and we try them in combination with an activator on latent HIV, you see that there's no synergizers or stabilizers of latency. So this is our control showing that basically we're seeing that non-modulators of noise are not enriched for synergizers or suppressors. What we did with these 3 latency promoting agents is that we we challenged them with potent combinations of activators. So we wanted to test how strong were they after we detected them. So here, this is a latent clone of HIV again with flow cytometry. You see that the untreated control, they're off when they're not treated. The activators are in red. These are all different types of activators and combinations of activators. And the activators, some of them are synergizers in themselves. So this is TNF, prostatin, trichostatin A, PMA, and combinations of them. And you can see that the red bars range from about 15% or 10% all the way to 50%. Now, in combination with the LPAs, the latency promoting agents, you get that they're pulling down this reactivation. So you can see that D75, the yellow bars, are really doing a good job at suppressing reactivation of latency across all of these different combinations of drugs. Now what are the mechanisms here? That's something that we're still interested in. But two of the strongest latency promoting agents, D75 and D35, were structurally and functionally related to inhibitors of the thioredoxin reductase pathway. It's a protein that's involved in maintaining redox in the cell of the host cell. And what we next wanted to do is test functional analogs of this pathway of thioredoxin reductase. And we found two additional thioredoxin reductase inhibitors that were also latency promoting agents. And they were called PX12 and tiopronin. So in this case, these are two full-length HIV latent populations, again, 9.2 and 15.4. You can see that untreated, they're not reactivating at all. With TNF is the dark gray dashed line. And you can see that after 24 hours, you can see that PX12 and tiopronin are both below the dark gray dashed line for 24 hours. You can also look at the 48-hour mark, and they're also below, they also suppress reactivation of latency. Okay, so let's change gears. How does this relate to brain? So these decisions are also occurring in the brain. It turns out that HIV is infecting multiple host cell types. It doesn't only infect T cells. It infects monocytes, it infects macrophages, microglia, and astrocytes as well. So these decisions that I'm describing to you are occurring in the brain as well. And we're interested in the monocytes and latency in the monocytes, the circulating monocytes, the monocytes that are crossing the epithelium and differentiating into macrophages and further infecting the macrophages and latency in macrophages. So this latent reservoir that I'm describing to you is very diverse, and it poses a challenge to treatment. So that's where Alexandra Blanco from my lab, she steps in, and her focus is HIV decision-making in diverse host cell types. She's working on HIV latency in monocytes and macrophages, and published a paper this year in Viruses on this topic. What she did is she established an in vitro model of HIV latency, a monocytic latency model. We call it the TLAT because it's based on a THP1 cell line. So we take the full-length HIV construct that has a delta envelope and GFP. We use lentiviral delivery and a low multiplicity of infection to infect naive THP1 monocytes. And then we do a GFP negative sort. So that way we get the latent cells and we reactivate those cells. So we use FACS sorting to get the GFP negative sorts, and then we want to find out which cells were the latent ones. So we use an activator such as TNF alpha in order to reactivate the cells, and then we do FACS sorting again into a plate, 96-well plate, single cell FACS sorting, and we grow out those populations to create a clonal population of a monocytic latency library. And then we can characterize this latent library of cells. So in total, Alex's library of focusing away from T-cell latency. Now we're interested in monocytic HIV latency. Her library consists of over 60 clones. On the left are the untreated clones, so you can see some of them have some low basal levels of expression, but a majority of them are off. With TNF activation, we have between 5 and 10% on for the most part, but interestingly, some of the integration sites really blast. There's hyper reactivating to above 40 or 50% on, and we're interested in all of these different behaviors. But what we did to start off with is we just took a single clone that reactivates at a low level, and we were interested to see whether chromatin modifiers and activators, in this case, these are the same ones that I showed you earlier that were the noise enhancers and the activators that synergized in the T-cell latency model. So on the right-hand side, we have JERCAT T-cell latency model. You see that the control is off. With TNF gives you about 15% on. The HDAC inhibitors by themselves do not reactivate this clone of T-cell latency, but in combination, the two synergize very strongly. And when Alex did the same experiment on a monocytic cell latency model on this specific clone, she got the same result. So you see that the purple bars are the HDAC inhibitors by themselves. In combination with the activator, you get synergy. So we see conservation of the response to a cocktail drug treatment between two different host cell types that are harboring latent HIV. What about the latency promoting agents? So this is some recent results. Alex took 3 of those latency promoting agents, and by themselves, you see that the control, this is now the monocytic HIV latency model, you see that by themselves, D106 and D35 are about the same as the control. The dashed bar is where the activating drug is. TNF is at about 4.5%. And then you can see that D106 and D35 suppress reactivation from what we can tell in this experiment. So we're optimistic that some of these latency promoting agents are also working across host cell types. And you can imagine for therapy and treatment in a diverse pool of host cell types, that'll be of importance, whether you're targeting only one cell type or multiple cell types, and for biasing latency. So in the time that I have remaining, I want to end with our stem cell work. So I'm going to shift gears to stem cells. Now, stem cells can for those that don't have that are not familiar, stem cells can self-renew and differentiate into any cell type in our body, making them attractive for tissue engineering and disease research. Their stem cell fate is determined by a core pluripotent gene regulatory network made up of 3 genes, SOX2, OCT4, and NANOG. This is a highly regulated core network of genes because in order to have reproducible and robust development to fluctuating environments and fluctuating signals, this core pluripotent network determines whether the cell stays in a self-renewal and pluripotent state or goes into a differentiation state between ectoderm, mesoderm, and endoderm. So in contrast to the HIV case where we asked the question, can noise enhancement be used to surpass a phenotypic threshold that was for reactivation, that was to go higher in expression levels? In the stem cell case, we have pluripotent and multipotent expression, which is higher, and upon differentiation with protocols and with certain differentiation factors, you get a loss of pluripotency. And what we're asking is, we're specifically interested in the cardiomyogenic differentiation and cardiomyocyte differentiation efficiency. And we're asking can we use a variable drug or growth substrate in combination with growth substrate in combination with differentiation factors to further enhance the phenotypic threshold crossings and get even more differentiation, and increase the differentiation efficiency. So back to the Waddington landscape, this would be similar to enhancing noise of the pluripotent state that's at the top of the hill and getting more cardiomyocyte differentiation at the bottom of the hill. So this is a project that's been led by Jason Wang from my lab on modulating pluripotent gene expression noise in embryonic stem cells. Specifically, we've been working on mouse embryonic stem cells to enhance cardiomyocyte differentiation. So developmental genes are coupled to cell cycle progression, and differentiation is initiated from the G1 phase of the cell cycle. And cell cycle modulation has been reported as a source of gene expression noise. So our hypothesis is that controlling cell cycle in pluripotent stem cells can modulate the noise of pluripotent gene expression, leading to enhanced cardiomyocyte cellular differentiation with differentiation factors. We're currently using CDK1 and CDK2 inhibitors to arrest cells, mouse embryonic stem cells, in the G2 phase of the cell cycle. So we can show that we can bias cells in the population into G2 and away from G1. We can also do antibody staining and flow cytometry and show that all three, SOX2, OCT4, and NANOG, display enhanced noise with flow cytometry. You see here on the left, the cyan or the light blue is one of the cell cycle modulating treatments, and the black distribution is the untreated control. And here, looking at the noise, the coefficient of variation squared versus the mean, the untreated control is the black square, the small black square here, and then an assay of several of these cell cycle modulators all enhance noise. So shifting the G2 phase, we're getting noise enhancement. And this is the next steps in this project is to test whether we're getting increases in cardiomyocyte differentiation efficiency and what are the implications for organoid, cardiovascular organoid formation, function, and development. This just shows that we're truly getting enhancement of noise in SOX2, OCT4, and NANOG. So I showed on the previous slide just the NANOG data, but we're getting noise enhancement compared to the untreated control in all three pluripotent factors. So we're cautiously optimistic that hopefully we'll have some results next time that we'll present on this, that we'll have some more results on the differentiation. But so far, we've been doing the noise modulation there. So I'm at the point of my summary and acknowledgements. I want to just summarize that I've shown you that quantifying gene expression noise requires single-cell measurements and is overlooked by measuring population ensembles for mean expression levels alone. Detected noise modulators targeted episodic transcription and HIV-positive feedback. These are two drivers of a stochastic viral switch of HIV decision-making to surpass phenotypic threshold crossings and bias latent stability. Noise modulators synergized or suppressed reactivation of latency, while non-modulators of noise had no effect. I showed you that, I think, only in the second noise drug screen, but not in the first noise drug screen, but we have the data for that as well in that first noise drug screen. And then latency reversal agents and latency promoting agents showed responsiveness to both monocytic and T-cell models of HIV latency. So this has implications for potential future therapies and therapeutics with regards to the various anatomical reservoirs of HIV. HIV in the brain, HIV in the lymph tissue, and bone marrow, and in other reservoirs where HIV resides. And then finally, cell cycle modulators enhance pluripotent noise towards controlling differentiation efficiency, organoid formation, and patterning. So this work takes a team of motivated and dedicated scientists, and I'm very happy and lucky to work with all of them. The graduate students that I've mentioned are here from my lab, as well as our undergrads that are working alongside them, our funding agencies on campus and off campus, some resources, as well as our collaborators that are on and off campus. I'd like to thank them, and I'd like to thank you for your attention today, and I'll take any questions now. Yeah.
So I have a question, kind of going back to when you were discussing noise suppression and enhancement. I'm still trying to grasp all of this, but my question is that when you suppress and enhance the noise, is there any type of compensation of noise that occurs in the system?
Yeah, yeah. So the question was if there's any compensation of noise in the system, it's not an easy answer, but noise does propagate in the system. There's noise that propagates in the network, and we're interested in those sorts of problems as well. And I alluded to in the very beginning that there's global resources that are kind of like pumping in noise at different stages of the gene expression that are just global resources that are fluctuating as well. So there's all different sorts of resources that are constantly kind of like a synchronizer that the noise is going up and down in the system. Probably, so for example, some of these noise enhancers that we had are not specific to our drug screen target. So some of them have, even though they're FDA approved, for example, some of them are anti-cancer drugs, and they probably are affecting many gene targets. So we're affecting noise in many places. So compensation is a tricky one. Compensation is probably related to if you have regulation that's occurring in a network. But if the gene circuit is isolated, we would like to get to the point where we're affecting noise in a very isolated way. But right now, I imagine we're also affecting noise in many different locations. I hope that answered your question.
Yeah, that actually does. That's very helpful. Thank you.
Great. Any other questions? Yeah.
So in your HIV studies, for instance, you did a few photon cells. Do you consider a normal cell, for instance, compared to the latent cell, whether those nice modulators that come for the ones that you need that they were become or chromatin modulators, whether also affecting noise in the normal CD4 cell compared to the latent, to the cell that will be infected and it's latent?
Yeah, so that's a really good question because we never actually measure the noise in the latent cell. In the latent cell, we only hypothesize that the noise will affect and bias the latent stability, but we never actually directly measure the noise. We can't measure the noise in it because the latent cell is off. But we actually, even if we got it to turn on, we never measure that the noise is also carried over and affects the gene expression there. So it is a kind of a two-stage process. It's kind of based on theory and the driving hypothesis. We go from an active expressing either mini promoter-only system or mini feedback system to the full-length latent virus. And that's a good question. We only detect the noise modulators of the active gene expression state. Yeah. Yeah.
You talked about some mechanisms for the latency promoting agents. Have you found any for the latency reversal synergizers? So you talked about the cytotoxic effects that they were similar.
Yeah, so mechanisms for the latency, so we have a lot of those latency reversal agent synergizers, and the mechanisms are pretty diverse. I think we have them clustered by mechanism of action in some supplementary table, but there are a lot of anti-neoplastics, a lot of anti-cancer drugs tend to be there. I can't think off the top of my head of what other mechanisms came up, but mechanism of action is a good question. It's definitely one we want to investigate further. Yeah. Yeah.
So, for example, mostly we use couple the HIV gene to influence the protein, for instance, and that will read out. And the question would be like, if you do the latency promoting agents, do you consider looking at the entire transcriptome of the cell itself, whether they are actually suppressing the transcription activity of the cell, the entire cell, and not only the HIV insect?
Yeah, so that kind of relates to propagation of noise and questions of off-target effects, and that's a great question. I think something like doing single-cell RNA-seq or doing some transcriptome-wide measurements can give us more insight as to what else is being affected. There aren't many papers that have done that. I can think of maybe just a couple that have shown kind of transcriptome-wide noise modulation. We haven't done it in this context, but that would be a good next step in seeing what sort of effects these drugs are having if we would take any steps further. We also haven't tried patient latency in patient models of samples. So there is some future work to be done. Yeah. Thank you. Yeah. Any other questions? Yeah.
Have you checked how long your fluid modulator efficacy? So I'm asking about pharmacokinetic effect.
You mean how long the duration that the drug is affecting the system that we're targeting?
Yeah, I know.
So like how long does the noise modulation occur for?
Mm-hmm.
So from what we can tell, from what we've seen, as long as the chemical hasn't degraded, it's still active and it's still working.
Okay.
Yeah. So we haven't necessarily done time-dependent noise modulation in that way, though. We typically kind of limit our experiments to 1 to 2 days, and that's enough to capture enough data and then transfer it over to the latency model.
I thought that your findings can really impact the for HIV. Are you talking to any farm to kind of reformulate the anti-HIV drug?
Yeah.
Basically delivering your fluid modulator.
Did you say farm as in like any companies? Yeah. We haven't taken it to the next step. We still have questions about mechanism that we probably have to get at, and we still have to prove that this actually maybe works in a patient model or with patient samples. I think that would be a nice next step because we're still only working. So even the primary cell data that I showed in the first noise structuring, I didn't show the second noise structuring also has primary cell data. But even in those cases, those are very that's the very first step of proving that these drugs are actually controlling latent stability beyond the cell line. So there's still more work to be done. But if there are companies out there that are interested, I'm happy to talk to them.
I did.
Yeah.
Yeah, I thought that maybe you can make your humanoid and test your fluid modulator for HIV.
Yeah, that could be interesting. Yeah, that could be interesting to see if it also carries over and it carries over between different systems like that. Yeah. Yeah. Any other questions? Any in the chat or in Zoom?
None from Zoom.
None from Zoom, it's quiet. All right. Well, then in that case, thank you.